Welcome back friends, welcome back in another video tutorial. So today we will going to talk about the functions of pentose phosphate pathway. This thing we have already talked. So today's topic is function of pentose phosphate pathway. So the first thing that we have seen formation of ribose 5-phosphate in the non-oxidative phase, right? That was the conversion from ribulose 5-phosphate into ribose 5-phosphate via isomerization reaction. Okay. Now this ribose 5-phosphate is important precursor for nucleotide synthesis and coenzyme synthesis. So from pentose phosphate pathway, we can prepare nucleotides and coenzymes by producing ribose 5-phosphate, right? Then there is a 4 carbon containing sugar present in the non-oxidative phase of pentose phosphate pathway, that is erythrose 4-phosphate, is useful for aromatic amino acid synthesis and vitamin B6, that is pyridoxal synthesis. Now, another important thing that various intermediate in pentose phosphate pathway are useful for production of ATP. For example, glyceraldehyde 3-phosphate from the pathway can enter the 3 carbon stage of the glycolytic pathway and produce pyruvate and ATP. Okay, that means you can understand that all the intermediates in the pentose phosphate pathway are somehow involved in production of some important biomolecules, right? So now the most important thing that pentose phosphate pathway produces is the NADPH. Okay, we have discussed that in case of oxidative phase, 2 molecule of NADPH is produced, right? And these 2 moles of NADPH is very important for many reductive biosynthetic processes, detoxification processes, oxidative defenses, reactive oxygen species generation through the activity of NADPH oxidase. Not only that, also in the synthesis of many important biosynthetic processes, NADPH act as a electron acceptor. For example, synthesis of fatty acid, synthesis of steroids, for example cholesterols, bile acids, steroid hormone, synthesis of sugar in the Calvin cycle, and synthesis of carotenoid. All these things, NADPH act as a electron acceptor. Now, one important thing that we also know that NADPH is electron acceptor and NADH is electron acceptor. That means what? Are they metabolically interchangeable? So answer is no, because the NADPH uses metabolite oxidation for reductive biosynthesis, but NADPH uses those free energy to produce ATP, right? That is why all the NADH involving steps are most of the cases oxidative phosphorylations, right? We have seen this kind of thing in multiple times when we have learned about glycolysis or Krebs cycles. So that is the thing. Not only that, conversion of ribonucleotides into deoxyribonucleotides also requires NADPH as a electron acceptor. Now, NADPH is also important for many kind of detoxification processes. One example is cytochrome P450 mediated detoxification of xenobiotics requires NADPH re-reduction. It is mainly accomplished by NADPH dependent cytochrome P450 reductase. What they do, they transfer electrons from NADPH to FAD-FMN electron transport chain. Then it transfers those electrons into the heme moieties of cytochrome P450. Okay, that is the thing. And it is also important in maintenance and regeneration of various antioxidative systems, for example glutathione, thioredoxins and superoxide dismutase. These things requires NADPH as a reducing agent. Now we will going to see a very important example, that is what happens if the first enzyme of oxidative phase of pentose phosphate pathway cannot work or deficiency of glucose-6-phosphate dehydrogenase occurs. So if glucose-6-phosphate dehydrogenase cannot be produced, then obviously there will be no synthesis of NADPH, right? Because the biosynthetic pathway cannot move, because we have learned that glucose-6-phosphate dehydrogenase mediated step was the committing step, right? So if committing step cannot occur, then obviously there will be no movement in the pentose phosphate pathway, right? So obviously there is no doubt that NADPH will not produce, right? And as NADPH is not producing, it will hamper many reductive biosynthesis processes and also lipid biosynthesis, right? Because just we have learned that NADPH acts as an electron donor in synthesis of fatty acids, right? So now we are here considering a situation that is, we all know about RBC, right? That is our red blood cell. So reduce glutathione. Now the question is, what is glutathione? Glutathione, remember, a biologically active tripeptide. Tripeptide means it is consist of 3 amino acid, and they are glutamic acid, cysteine, and glycine. Okay, and interestingly, glutamic acid and cysteine are joined via glutamic acid's gamma positions carboxylic acid, right? Because we all know that in case of glutamic acid in the R group there is a COOH, right? This COOH will going to form peptide bond with the NH2 of cysteine. Okay, that is the idea of glutathione. So this glutathione in its reduced form is very important for maintaining the integrity of RBC. Okay, for example, this reduced glutathione, I am mentioning this reduced glutathione as RGSH. This reduced glutathione is important for removing H2O2, which is a toxic byproduct of various oxidative processes. Okay, this H2O2 can react with the double bond of the fatty acids which are present in the membrane of the RBC, because we all know membrane are made up of what? Fatty acids, right? Fatty acid residues. So by reacting with that double bond, H2O2 can form organic hyperoxides. Okay, and this organic hyperoxide facilitates the lysis of that particular cell, which is RBC in that case, and facilitates hemolytic anemia. Okay, so what is going on? As NADPH is not producing, okay, reductive glutathione somehow is not present. Okay, and as a result, H2O2, which are the toxic byproduct of various oxidative processes, cannot be producing. Sorry, cannot be removed. Okay, so as it cannot be removed, it will going to react with the double bond of the fatty acid present in the membrane of RBC. Okay, and this reaction facilitates the production of organic hyperoxide. Okay, and this organic hyperoxide will promote the premature lysis and hemolytic anemia. Okay, okay, so that is the idea. So now what glutathione will do, let's learn. So we will going to use this picture to understand what glutathione will do. So reduced glutathione will donate its electron to H2O2 and convert it into water with the help of enzyme glutathione peroxidase. Okay, and as reduced glutathione is donating its electron, it becomes into its oxidative form of glutathione. Okay, so that is the idea. Now as it is now in its oxidative form, it should be recycled into its reductive form, right? And this is accomplished by glutathione reductase. Okay, the NADPH which is generated by pentose phosphate pathway will going to donate electrons in presence of glutathione reductase to the oxidized form of glutathione and convert them back into reduced form of glutathione. Okay, that is how the entire thing is going on, and you can understand how important NADPH is. If NADPH is not produced, then after donating electron, the oxidative glutathione will not be converted back into the reduced form of glutathione, and as a result, further H2O2 if produced in the cell, it will not be converted into H2O, right? And as a result, it will facilitate premature cell lysis and hemolytic anemia, right? So how important NADPH is, you can understand. Now we will going to revise this once again. What I have told you, that is in RBC, let's consider that this circle is RBC. So RBC's membrane integrity is maintained by plentiful supply of this thing, that is reduced glutathione. Okay, if reduced glutathione is not present, then H2O2 that are producing for various oxidative processes will going to react with the fatty acid residue present in the membrane of RBC and produces organic hyperoxides. Okay, that will ultimately facilitate premature cell lysis and hemolytic anemia. Okay, which is not a good thing. Now, why reduced glutathione shortage is occurring? Because of the absence of NADPH due to glucose-6-phosphate deficiency. Okay, what NADPH will contribute? NADPH will going to donate its electron to the oxidative form of glutathione in presence of glutathione reductase and convert them back into reduced glutathione, so that glutathione peroxidase enzyme will use this to convert H2O2 into water. Okay, so that is the entire idea, right? So that is how deficiency of glucose 6 phosphate dehydrogenase will promote premature cell lysis and hemolytic anemia. And what is the role of NADPH here? You can understand, right? So that is the ending of our topic of pentose phosphate pathway. Hope you have enjoyed this video very well and understand what I have learned to you, right? So now in the next video we will going to talk about ED pathway. So till then, bye bye.